Hello everyone, and welcome to our online lecture series for Bio 2. Today, we continue on with our exploration of inheritance and Punnett squares, with today's entire lecture being focused on problem solving different types of Punnett square problems. This means that we're going to expand on our understanding of, first off, the Punnett square with a pedigree, and then also expand on our understanding of different modes of inheritance, meaning that we're going to break that dominant recessive paradigm. But before we do that, let's take a look at how dominant and recessive play out in this pedigree analysis. So, some of you who are familiar with animal breeding may have heard the term pedigree before. Basically, it means a family tree where you ascribe some genotypes to the individuals in each generation. So just like we were doing with our Punnett squares, we have a parental generation, a filial 1, a filial 2, a filial 3, so on and so forth. But rather than just keeping within the family, pedigrees allow for individuals who are unrelated to come in. So for example, this individual comes from some other group, is connected to the pedigree elsewhere, not to the original parents, but because they mate with this individual here, they contribute genetics to the third generation. And in this way, we can kind of more accurately keep track of what's happening in family history. Because ultimately, one of the greatest uses of genetics and pedigrees is to inform your own family medical history. So that way you can understand what things you should be particularly concerned about. So that way you can live a happier and healthier life. In this case, we have numbers indicating different individuals, shapes indicating their gender, with males being squares and females being circles. And then we have individuals who are unaffected by the disease as yellow and affected by the disease of alkaptonuria, quite the mouthful. But for the purposes of this example, the physiological effects, which alkaptonuria causes your urine to turn dark because of inability to metabolize different waste products, those physiological effects are not important. We're instead focusing on the inheritance patterns. So we need to take a look at what are the possible genotypes for parents 1 and 2. Well, we know that they produce an affected child, meaning that at the very least, one of their 2 genotypes for each of them has to be a little a. And I know I said for all problems that I'm assigning, please keep to a capital or a letter for the gene with subscripts for the alleles. But because we're doing this example out of the book, we're going to keep to the capital and lowercase. It's good to be able to flex as to whether you're working with different nomenclature systems. But again, please, for your own work, it is a lot easier to try to keep track of these things with a gene letter and then allele subscripts. So, the father with the square was affected, and a child with 1 capital letter, even though this is not their child, but an individual with a capital letter is unaffected. So we're likely looking at a mother that's heterozygous and a father that's homozygous, recessive for alkaptonuria. When these 2 individuals mate, they produce 3 children. 1, 2, and 3. They produce a girl that has it, a boy that does not, and a girl that does not. We see no variation in genders here, so boys and girls can have it. We have both circles and squares having it. So it will not be something like sex distorted here. It's a simple dominance and recessive model. So what is the genotype of our second individual here? What are the possible babies that could have been produced from a Punnett square of a big A, little a, crossing a, little a, little a? Well, it's either het or homozygous recessive. And since we know that this person is unaffected and has a big A, we know that their second allele has to be a little a. Cool. Continuing on with this tree, we could solve for this individual's genotype as well. They mate with one of the people connected to our family tree who is homozygous recessive, and they produce a homozygous recessive baby. So this actually parrots what we saw up here, where you have an affected mating with an unaffected, producing a homozygous affected. This means that this individual has to have been heterozygous. For our last unknown genotype, we have a heterozygote mating with a heterozygote, one from the family tree, the other brought in. The progeny here, you have one affected individual and another individual who is unaffected. And you want to know what their second allele is. Well, if we draw the Punnett square for this cross here, we have a heterozygote crossing with a heterozygote, which, hopefully from last lecture, you know can produce 3 different genotypes. Homozygous unaffected, 2 possible heterozygotes, and a single homozygous recessive or homozygous affected. That means that this individual has the option of either being AA, capital A, or heterozygous. And if we know that they carry one of the big A alleles, we know that they are not homozygous recessive. Therefore, they have some reduced chance of being some greater chance of being one of these 3 genotypes. And since there are only 3, that means they have a 1/3 chance of being capital A, and a 2/3 chance of being heterozygous. And so in this way, we can start to build and inform our pedigrees. Now, pedigrees are very important in uncovering one very particular mode of inheritance, and that is traits that are X-linked. Meaning that these traits are found on the X chromosome. So, building on our knowledge from before, here we're obviously talking about the gender determination system of XY, not ZW though. It's fair game as well. Things will flip around there. In X-linked traits, you can have traits that are either dominant, meaning that you only need one allele in order to express it, or traits that are recessive, meaning that you cannot have the other allele if you want to see it. But there's an additional complication to working with X-linked traits, and that is the fact that not all genders have the same amount of X chromosomes. We know, for example, that females are XX. That means that they will be diploid. And we know that males are XY. They only carry one X chromosome. The Y chromosome does not carry the same genes. In fact, it has orders of magnitude less. This makes our males hemizygous, because they will always be haploid. I'm putting quotes in my hands here. So they'll be haploid in their genotypes because they only carry one of the 2 possible alleles, because their chromosomes don't each carry one. There's structural variation across the genome. So in X-linked traits, you're looking at how individual genotypes manifest differently in males and females. For example, in a trait that is dominant and X-linked, a female needs to have it only once in order for it to be showing. So in this case where you have 1s and 2s, actually, we'll now match the letters, capital and lowercase letters here since they look very different. A capital B means the disease that we're looking at, and the lowercase b means unaffected, meaning that this trait is dominant. This individual has it, and I'll indicate has with a plus, and then an individual that does not have it with a crossed out circle. So here you've got, in a female carrying one allele, having the disease versus not having the disease. How does this manifest in a male? Well, if we're looking at our male possible genotypes, an X with a capital B and a Y, well, only one allele, it's going to have it. And an X with a lowercase b and a Y, not going to have it. So we have one that's affected, one that is unaffected. So what happens if we cross these? Well, I'd like for you to be able to make all the mixes and matches here. Do some practice of combining all of the different genotypes. Only a couple different matches here. But let's go ahead and pull out one of these. Let's say that you have a female that is affected, but heterozygous, and a male that is unaffected. What progeny can these two make? Well, one of them will be a female that is affected. One of them is a female that will be unaffected. One of them will be a male that is affected. And the other will be a male that is unaffected. So despite the fact that you're looking at one locus, the fact that this is X-linked means that you get 4 different outcomes. One affected female, affected female, non-affected female, affected male, non-affected male. Let's see how this differs when we look at recessive traits that are X-linked. And in this one, we have a very well-known example of hemophilia. I actually have a second cousin with this. Nowadays, hemophiliacs can live healthy lives. We figured out the medicine necessary in order to supplement their blood, so that way they can clot, because hemophilia is a clotting disease where you are unable to form scabs. This is hugely problematic as internal bleeding will continue. But hemophilia is the disease that is carried on the X chromosome, and it encodes a broken version of that clotting factor. So, in a female, let's say that X big H means normal or unbroken, and X little h is the hemophiliac. In a female that is X big H, X little h, because this is recessive, this individual would be unaffected. In order for a female to be a hemophiliac, they need to be homozygous. However, when they have a single allele for hemophilia, we call them carriers. So let's go ahead and mate a female carrier with an unaffected male. Female, carrier, unaffected, male. And let's see what their progeny types are here. X big H, that is a unaffected, not a carrier female. X big H, X little h, this would be a carrier female. X big H, Y, an unaffected male, and X little h, Y. This individual is affected. And in this way, we can see how it is more likely that males express the recessive X-linked traits because they don't need 2 copies of it, they just need 1. Females, having that backup copy, make them genetically a bit more resilient to these X-linked diseases. And in the case of hemophilia, the reason why it is so well known is that hemophilia ran in the family tree of Queen Victoria. So looking at this pedigree, and back in time, we know that Victoria was a carrier of the disease. And she mated with Albert, who did not have the disease. They produced a large number of children. And what you will see is that they had children, particularly males, that were affected. Victoria passed the hemophiliac allele to her daughter Alice of Hesse. Alice then mated with an unaffected male and passed it to her son Frederick. The Duke Leopold of Albany had the hemophilia allele, as well as Victoria's daughter Beatrice, who then passed it on to Maurice. And obviously, these lines where they indicate a single line drawn, they're excluding the mating pair there. There is mating there, but as you can imagine, the mating patterns of these giant medieval royal families were quite complicated. So here, this illustration just goes to show you a pedigree of an X-linked recessive trait like hemophilia. And so that covers our first of several different types of inheritance patterns, both looking at them from a pedigree standpoint, as well as individual Punnett squares, looking at dominance and recessive inheritance on the X chromosome. Next up, we've got an inheritance pattern that cares a fair bit more about molecular manifestations, or how it looks on the cellular level. These 2 patterns are those of codominance and incomplete dominance. Now, I will go ahead and say that these 2 are far from my 2 favorite names of a system, and even my favorite genetics textbook actually calls out these 2 as not really that different. But still, I will show you how they kind of manifest slightly differently in order so that way you understand how they get passed on in the way of a Punnett square. Codominance is defined as both alleles expressed, meaning that it's not possible to be completely masked in a heterozygote. One good example of this is our A, B, O blood groups. So the way this works in ABO blood groups, actually just move this down here, is that your blood, any nucleated cell in your blood, has this type of blood type. The A, B, and O are different types of glycoproteins, or sugar signaling proteins on the outside of the cell. So here you've got a cell. Oops, I like the consistency of copy-paste. And each of these cells are from different individuals and will carry 2 different alleles. So our gene is going to be the blood type gene, so we'll indicate it with a B. And then our 3 different alleles, because you have now a case of multiple alleles. So this is kind of a 2-for-1 in codominance. Put a little asterisk up here for multi-alleles as well. You have the blood type A, you have the blood type B, and then you have the blood type O. And A and B are codominant, and O is recessive to both of them. So the way that this works is if you have a blood cell that is BA, BA, this cell will go ahead and make proteins on the surface of the cell that have A receptors, and only A receptors. That means this cell will float around going, hey, hey, I'm A, I'm A, I'm A, I'm A. If you're anything but A, I'll pick a fight with you. A cell that has the blood type B and is homozygous for it will also have cell receptors that are going around screaming, hey, I'm B, I'm B, I'm B. If you're anything else, I'm going to pick a fight with you. And blood type O does not make receptors on the outside of the cell that scream anything. They're just quiet. So what happens when you mix a cell that is BA, BB? Well, the A makes a functional receptor that gets to the outside, and the B makes a functional receptor that gets on the outside. And so you have cells that are AB. These have both of those different receptor types. This individual's blood type would therefore be considered AB. If they receive blood that is A, they are cool with it, and they can receive blood that is B and be cool with it, and they can receive blood that is O and be cool with it, because they're looking to either match with the A's, match with the B's, or hang around the quiet O's. Instead, what happens when we have a blood type with a heterozygous O? Well, in this case, let's say that it's a AO blood type. This cell ends up looking like it has an A receptor and an empty B glycoprotein, or an empty second glycoprotein. This individual's blood type would therefore just be A. And in this way, this individual could receive A blood, matches just fine, could receive O blood, it's not picking a fight, but could not receive B blood. And so in this example, you see that there are expression patterns. It's not that they're different genes or something else is going on. You just have a case of codominance where the allele for blood type A gets to the surface and is made and lets itself be heard. and the allele for B makes a functional protein that gets to the surface of the cell and lets itself be heard. In this way, the blood type AB is known as the universal recipient, meaning that you can throw in any kind of blood into an AB individual and they'll accept it because they recognize either A or B or AB or O. They're not particular to any of those. In contrast, if you have an individual that is just BO, meaning homozygous for the recessive state, in this case, they make no functional outside protein. So if the other components of their blood encounter either A blood or B blood or AB blood, they reject it, they fight it. They think that this is not here for peace, they think that this molecule is here for war, meaning that it's a foreign invader, and it triggers their immune system. So BO can only receive BO blood. But because BO blood is not picking a fight by making any particular receptor on the outside, it is known as the universal donor. And so hopefully that connects to some of your previous understanding of genotypes and blood and molecularly what's happening on the cell. So let's go ahead and do a Punnett square here, where you have an individual that is homozygous A, an individual that is heterozygous B. And if they're heterozygous B, you know that they have to have an O allele because they're not expressing the A, and they would be homozygous B if it was two little b's. This is not an X-linked trait, so we're using it just on autosomes like we were before. You'd get one individual that is blood type A, B, another individual here that is blood type A, B, an individual here that is blood type A, but heterozygous, and an individual here that is blood type A and heterozygous. So two different genotypes, a 50% chance of being blood type AB and a 50% chance of being heterozygous blood type A. So hopefully you can begin to appreciate how important it is to understand these different terms. I'm already throwing around heterozygous, homozygous like they're hotcakes, right? It's in every other sentence while we're discussing this. So please, if you need to, slow down the lecture, make sure you understand this vocabulary, so that way the questions that I ask you on your quizzes and final and midterm and final exams make sense to you based on the vocabulary that we're using. Okay, so with codominance, or a system where both alleles are expressed because it's not masked in the heterozygote, so in cases where you have a heterozygous genotype, the A does not prevent the B from getting to the surface, and the B does not prevent the A from getting to the surface. Both of them actually make their products. Let's take a look at how it's slightly different in incomplete dominance. In incomplete dominance, you would instead have a system where one allele is unable to completely mask the other. The best example of this is in flower color, where you can have three different systems of three different colors of flower characterized by the presence of an intermediate. You can have flowers that are red. You can have flowers that are white. And you can have flowers that are pink. Flowers that are red, let's go ahead and define our loci. So F for flower, the red allele is FR, and the white allele is FW. A flower that is red will have the genotype FR, FR. Let's go ahead and move our discussion of flowers to below the box. Sorry about that, y'all. So that way I have enough room, not crowding things out. An individual that is FW, FW would be white. And an individual that is FR, FW is pink. And again, on a molecular scale, let's take a look at how this works. Because we're drawing plant cells, we're drawing them in a square. Animal cells are circular, or generally more curved or rounder. Plant cells have a plant cell wall. So we draw them as rectangles, even though looking at them under a microscope, you know that they are not all these neat little squares. In a plant cell that is red, let's go ahead and say that you have 2 or 4 receptors, 2 or more places to put a functional protein. And in the case of a red, actually, you have lots of places to put a functional protein. In the case of a red, you're going to line the outside of the cell in a whole bunch of red. In the case of a white, you line the outside with a whole bunch of white. This is not working out that great. Can't see that? Interesting. Okay, well, we'll do it in gray. There we go. And so the only proteins that are being expressed are those different pigments. Well, in the case of a pink flower, you will get some red being made, and you will get some white being made. Now, because both of them are getting to the surface, both of them in this case are being expressed, you now get this situation where the red cannot mask the white completely, nor can the white mask the red completely. From a bird's eye view of really, really far away, because keep in mind that these are cells, so they're very tiny, that red next to white, that red next to white, that red next to white, well, that ends up looking pink. And so in this way, it's not 2 different or 3 different alleles here. You just get a blending of the heterozygote. This can be caused either by both expressed but neither mask, on a molecular level, or you can get a situation where the red makes a functional protein and then it gets to the surface, and the white makes a protein that never gets to the surface. It stays on the inside. In this way, white might be broken, but the fact that it's not manifesting against the surface means that it's not taking space there. Your cell won't make more red to take the place of the white if it's not there. It makes, let's say it makes 10 red and 10 white off of each copy of the allele on each of the chromosomes. And therefore, the heterozygote individual will look less red than a homozygous red individual and will therefore look pinkish potentially. So you can get pink either by both expressed and neither masking, or one expressed, other not. Therefore, not masked either. But in both cases, you're getting the situation where the intermediate is manifesting as a different appearance. You'll notice how that is fairly similar to codominance, right? You might get a situation where both colors are expressed, like case 1, and now we have a pink intermediate, just like you might consider an AB some sort of quote-unquote intermediate. But because AB functions more on a qualitative, do you have A or do you have B or do you have both? There really is no blending. They're expressed as separate units. In the case of flower color, on the very surface of the cells of the flower, you can see that there is red pigment and that there is white pigment, but when you zoom out on it, it just looks red, white, or pink to you. So let's do a quick Punnett square for this. Let's go ahead and say that we have a pink flower cross a white flower. Well, any pink flower you know will have to be heterozygous, and any white flower you know will have to be homozygous white. This means that the progeny that they can create are a pink flower, a white flower, a pink flower, and a white flower. Our pinks and our whites. Now, interestingly, if you then selfed the pink flowers, you then could get a red-colored children. So in that way, you could potentially recover the phenotype of being red from flowers that are pink. So hopefully, codominance and incomplete dominance make sense to you. Again, we covered a little bit about multiple alleles in our discussion of codominance. Take a moment here to make sure that you can switch up some of the crosses. I might recommend changing the blood types from an individual that happens to be A and a heterozygous B to maybe a heterozygous A and a heterozygous A. What blood types can they be? Challenge your friends to different mix and matches. Same with the flower colors. Mix and match them. Before you head on, make sure that you understand this. All right. So, time to move on to our next type of inheritance. Fortunately, we're going to switch from something that's a little bit complicated, like codominance and incomplete dominance, to a much more simple mode of inheritance. This is either the mitochondrial or plastid genes. Plastid meaning your chloroplasts. Mitochondria being those powerhouses of the cell. Now, mitochondrial inheritance patterns are going to be important for determining human history things because we'll use mitochondrial haplotypes in one of our labs this semester. But the general idea here is that your mitochondria has its own genome. But that genome is always haploid. So that means you only have one type. So essentially, our 2-box Punnett or our 2x2 Punnett square is going to have fewer possible locations to add information. Now, when you get your mitochondria as a child, you are only getting them from your mother. Mitochondria are present in the egg. They are not donated from the sperm. This means that not only are you haploid for these, but you only get your mother's genetics. For your mitochondria. So, not only do we not have the second squares here, let's grab the bigger marker here. Not only do we not have the second squares here, but you don't even have the first square from your dad. Dad does not donate any genetic material, therefore your mitochondrial type will be directly inherited from your mom. Now, while this makes for a very silly Punnett square, imagine this on a long-term, evolutionary study for human migration patterns. You could tell which maternal lineage groups come from. And as we'll talk about later, you can even trace back all of humanity to one particular mitochondrial haplotype shared by a group of mothers way back, to the point where all Homo sapiens as a species has a single mitochondrial mom. Now, we very cleverly have named that individual or that mitochondrial haplotype mitochondrial Eve. But before we get to talking about how we got there and what that means, looking at it a little bit more detailed DNA level, you need to understand that mitochondria are inherited maternally and that plastids are inherited maternally. So the same things that go about our mitochondria, meaning that they are inherited just from your mom, your dad's type disappears. It's not added on into the children. You instead have, you additionally have the same thing occurring in the chloroplast. It's also got its own genome. It's also got its own DNA. And it also is only inherited maternally. So you could tell which maternal line different groups come from. So definitely thank your mom for giving you that powerhouse of your cell. So hopefully fairly straightforward about mitochondria and plastids. Because next up, epistasis. All right. So epistasis means on top of control. And epistatic problems are always 2-gene problems, at the very least, because you are going to have one gene as the controller, and then one gene as the factory or the producer. And this oftentimes leads to 3 phenotypes, not 4. Because you remember in our dihybrid crosses of pea plants, you might have crossed pea color that was either yellow or pea color that was green for your first gene. And then at your second gene, you might have had flower color that was violet or flower color that was white. And you can mix and match these with yellow peas with purple flowers, yellow peas with white flowers, that's 1, 2, green peas with white flowers, that's 3, and green peas with violet flowers, that's 4. Those are your 4 phenotypes. In an epistatic question, you're instead going to get 3 phenotypes. Agouti, and in this example, agouti, the tannish color for our little mice, a black, and an albino. So let's take a look at this cross between 2 parents. Again, they don't note gender here because this is not epistatically controlled X-linked, thank goodness. I won't be doing that to y'all. Instead, we have epistatically controlled somatic or autosomal chromosomes. And so male and female are not particularly important. What you do notice that this is again a 16 square Punnett square, and that we're again putting one allele from each of our 2 genes on top of each box. So 2 letters for each row or column, and they have to be two different letters. And then ultimately, each of our squares is going to have 4, meaning that you are diploid at 2 different genes. So, how does inheritance of this work? When do we get different phenotypes? Well, you have your 2 genes. The A gene, where a capital A is your agouti coloring, and your lowercase a's are black. And the way this works is either you will produce some sort of tan-colored pigmentation with the dominant tan allele, or you will not produce any tan coloration, instead a pigment that absorbs all light, if you have 2 recessive copies at the agouti gene. So if I was doing this, I would have labeled them differently, but I'm going with your book here. So you have your agouti locus, and then you have your control locus, or control gene. If you have 2 capital C's, this means that you do make color, meaning that your color is going to be checked at your agouti, meaning proceed, go ahead, make a color gene. The same is true if you are heterozygous. But if you are homozygous, little c, no matter what color you have at your agouti locus, you never make any color. Therefore, you end up albino. This means that any haplotype or any genotype that has 2 little c's will always be albino. And there are our 4 albinos. And so, in this case where we have this dihybrid, meaning that there are 2 heterozygotes for color and control loci, you can see that there's this ratio of 9:3 to 4, not 9:3 to 3:1. Because rather than the different homozygous recessive at locus 2, or your control locus, rather than them separating out into 9:3 to 3:1, you take that 9:3 to 3:1 and group them together into your white phenotype. So we'll do, with your book having solved this one, we'll do another problem in this vein with our own labeling system. So here you have different types of hair color in humans. In us, humans. We are going to be looking at, and this is a completely made up example, so there's no real genetics underlying this, but we'll be looking at purple hair, gray hair, or to be distinct here, I should do silver, and then bald, or the inability to grow hair. Now, in terms of these 2 loci, you have the hair color locus that can be either purple, which is dominant to silver, and then you have the bald locus, where you either are not bald, making color, or you are bald, meaning that you make no hair. Fortunately, in this case, the not bald is dominant to the bald. Let's say, for example, that we have an individual with the genotype HP, HS, BN, BB. So a heterozygous individual. This person would first check whether they do or do not make color. So they have one gene for making no color, one gene for making color. Since one gene makes color, they can go ahead and make their color. Now, they have two possible alleles for making color. They can either make that silver color or they can make the purple color. Well, since purple is dominant, this individual is going to go ahead and make a purple hair color. Let's see what happens if we have an individual that is bald. Say that they too are heterozygous. This individual checks first their control that they are bald. Because their control tells them that they are bald, they never make anything in the hair color. So you might not even know what genotypes they have for hair color. So using these crosses, one individual that is bald and one individual that is purple, given the circumstance that you know that the bald individual has an underlying phenotype or underlying genotype of heterozygous at the hair color locus, let's do a big old Punnett square. So we'll call your bald individual your female and your purple-haired individual your male. Let's write out all of the genotypes on here. So looking at our purple female, we know that they can be purple paired with the not bald. Purple paired with the bald. Silver paired with the not bald. And silver paired with the bald. All four types. Now for our, I mixed these around. We will instead have it be, sorry about that, we will instead have it be the purple-haired female with the bald male. I tried to switch it around for gender stereotypes, but I just wrote them on the wrong side. Not particularly important here. On our male side of things, we will pair the purple with the bald and the purple with the bald. And then the silver with the bald and the silver with the bald. But you'll note that that repeats twice. So actually, let's just draw this out so that way we can see. The purple with the bald, the purple with the other bald, the silver with the bald, and the silver with the bald. Because these repeat, we don't need to fill them in. So we can expedite our writing of this Punnett square. And the ratios will simplify out. So let's go ahead and fill this in. Well, HP and HP, carry over this HP and this HP to combine to make that, and then carry over our balds. Bald not, bald definitely. Purple hair, that's actually bald. And then purple hair with a silver, but a not bald with a bald. And then purple hair with a silver, but double bald. Now, purple hair with a silver, with a not bald and a bald. Purple hair with a silver, but a double bald. Silver hair with a not bald and a bald. And then silver hair with a double bald. So now let's go ahead and color these in. This will indicate that the individual is purple-haired. This will indicate that the individual has silver hair. And what color should balding be? Brown. Maybe a darker brown. Or that will do just fine. Bald. So, which is it for our first genotype here? Well, I noticed that they have a not bald allele, so let's go ahead and check what colors they make. Well, they are purple, so they have purple hair. Here again, you have the balding locus, but you have two balds, so who cares what you make at the purple? This individual is going to go ahead and be bald anyway. For our next individual, we've got a not bald and a bald, so let's check what their hair colors are. A purple and a silver. This individual would therefore be purple. Next individual, two balds, who cares what's elsewhere? We are bald. Down below, you'll notice the pattern. Two little b's, two little b's. Let's go ahead and make those two balds right away. And then two cases where you have balding with silver. And purple with non-bald. So this weird situation where you had heterozygous at both, and then het for color with homozygous recessive bald, you end up getting a ratio of 3 purple to 1 silver to 4 bald. And if you carried it out and wrote out these other boxes, you would notice the exact same trend. But hopefully, this epistatic control makes sense whether you're looking at mice and agouti coloring or human hair color in completely made-up examples. Note that come time on the exam, you will be given some sort of question in the vein of likely coloration, but you should be looking for those 3 phenotypes with one gene controlling the different colors. It's a very common way to do epistasis. All right, hopefully that wasn't too bad. We'll end our inheritance lecture with our last possible Punnett square. And this one just ends up looking cool. Pay no attention to the giant square at the bottom, just yet. This type of inheritance is actually the most common type of inheritance, which makes it really frustrating that the book does not even include an example on this type. This type of inheritance is called quantitative, or additive inheritance. Because the idea with quantitative inheritance, is that there's a numerical game here. You have different genes. Let's go ahead and label them maybe gene A, gene B, and gene C, very cleverly named, I know, that then have different values that add a different amount to the overall height of the individual. Height is one of our greatest or most quantitative traits in terms of human genetics. Gene A might add an inch to height if it's got allele 1, and nothing to height if it's got allele 2. Allele B will add 1 inch to height if it's got allele 1, and nothing if it's got the second allele, allele 0. Same goes for C. So then my first question for you is, what is the height of an individual who has, looking at only their A and B. Oh, sorry. We'll do this one at a time. My bad. We will look at only one gene here, two genes in our 4x4, and then three genes in our 8x8. So an individual that is heterozygous for our one gene system, let's say that this is gene A, we're looking for an individual that's heterozygous. What is their overall height? Well, that equals 1 inch. Whoops, I have to find a way to distinguish my ones from random lines. 1 inch. What heights of children can they produce? Well, if they're heterozygous at both of these, you'll note that they can produce an individual that is A1, A1, A0, A1, A0, and A0, A0. If you're looking at this on a graph, you'll notice that they can produce, so this is the count, this is your height, going from 0 to 1 to 2, and on the left side you have your count of 0 individuals, 1 individual, 2 individual, and 3 individual. So how many individuals have a height of 0? Well, just the 1. How many individuals have the height of 1? Well, 1 and 2. And then how many individuals have the height of, so this was your height of 0. These two are your heights of 1, and this is your height of 2. Do you notice the shape of this? Additive traits follow a normal distribution. And most things in nature abide by this normal or semi-normal distribution. So in the same way, let's go ahead and shrink this down a little bit. You could put a Punnett square together and then a histogram, which is exactly what this is, for a 2-gene system where you have genes A with A1 and A0 and gene B with 1 and 0. Now, to expedite this process, rather than writing out a whole boatload of numbers on the top here, we could take a look at how they pair and calculate the height that they add. So if this is your genotype here, let's do your first, inner, outer, last. If I pair my firsts, A1 and B1, that gives me a height of 2. If I pair my inners, that pairs A0 and B1, that gives me a height of 1. If I pair my outers, that gives me a height of A1 and B0, height of 1. And if I pair my lasts, A0 with B0, gives you a height of 0. You can replicate this on the other side, a height of 2, a height of 1, a height of 1, and a height of 0. Then all you have to do is add the values in the square. So 2 plus 2, 4. 2 plus 1, 3. 2 plus 1, 3. 2 plus 0, 2. 2 plus 1, 3. 1 plus 1, 2. 1 plus 1, 2. 0 plus 1, 1. 1 plus 2, 3. 1 plus 1, 2. 1 plus 1, 2. 0 plus 1, 1. 0 plus 2, 2. 0 plus 1, 1. 1 plus 0, 1. 0 plus 0, 0. And so now, in this dihybrid, you are again not getting just 4 different phenotypes, but you're getting one phenotype with many different heights. So you have a height of up to 4 and all the way down to 0. You'll have 1 at a height of 0 and 1 at a height of 4. So that takes care of those two. How many 1s do you have? 1, 2, 3, 4. 1, 2, 3, 4. How many 3s do you have? 1, 2, 3, 4. Well, there's also 3 3s. Or 4 3s, sorry. And then how many 2s do we have? 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Again, notice that curve, notice that normal distribution in our histogram, where again you have heights on the bottom and counts on the side, just like you did in your genome size lab. So for our 3-gene system, I will leave this for you to fill in because it is great practice. But in the same vein, looking at height, you have 3 loci. Each individual is a heterozygous at all 3, meaning that the genotypes are A1, A0, B1, B0, C1, C0. This individual's height is 3. If I was to cross this with another individual of the same genotype, I'd like you to write out all of the different gametes. Well, pause the video here if you need to, otherwise I'm going to get to writing them out. And please do pause the video, don't cheat yourself of this. It is absolutely critical because you're no longer just following FOIL, you're pairing multiples here. So let's first pair all of the A1, B0s with all of the Cs. So A1, B1 with C1, A1, B1 with C0. Now let's switch over to pairing A1 with B0 with each of the Cs. A1, B0 with C1. A1, B0 with C0. Those 4 will be replicated when we look at our A0. So let's pair A0 with B1 and then each of the Cs. So A1, or A0, B1 with C1. A0, B1 with C0. Now that we're done there, let's pair A0 with B0 and each of the Cs. A0 with B0, A0 with B0 with each of our Cs. And let's calculate the heights of each of these. Well, this would add 3. This would add 2. This would add 2. This would add 1. This would add 2. This would add 1. This would add 1. And this would add 0. So you can now go ahead and fill these in on these sides of the Punnett square. A, 3, a 2, a 2, a 1, and let's make sure that I'm counting these right. A 3, a 2, a 2, a 1, continue on, a 2, a 1, a 1, and a 0. A 2, a 1, a 1, and, last but not least, our 0. Replicate that. 3, 2, 2, 1, 2, 1, 1, 0. And now for some math. We have a 6 here. 3 plus 3 is 6. This is our tallest possible individual, right? 3 plus 2 is 5. 3 plus 2 is 5. 3 plus 1 is 4. 5, 4, 4, 3, 5, 4, 4, 3, 4, 3, 3, 2. 5, 4, 4, 3, 4, 3, 3, 2. 4, 3, 3, 2, 3, 2, 2, 1. Next up, keep track of this. 5, 2, nope, no. 5, 4, 4, 3, 4, 3, 3, 2. Make sure my 2s and 3s look different enough here. And then our 4, 3, 3, 2, 3, 2, 2, 1, 4, 3, 3, 2, 3, 2, 2, 1. And last but not least, 3, 2, 2, 1, 2, 1, 1, 0. 64 possibilities, right? 8 by 8. Let's count them up now. We go from a range of 6 to 0. I'm going to zoom in here so that way I can write more accurately. 1, 2, 3, 4, 5, and 6. Let's count up how many of each of these we have. Let's see if I did it right. Well, you know that you've got 1, 0 and 1, 6. See how it's really difficult to be super tall or super short? Not a lot of likelihood for that happening. Now, in terms of 1s, we've got 1, 2, 3, 4, 5, 6 of them. In terms of 5s, if we're doing this correctly, we should have the same. 1, 2, 3, 4, 5, 6 of them. 4s. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 4s. 2s. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Whew, I didn't mess it up. And 3s. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. 20. Double check yourself. Does that add up to 64? 1 plus 6 is 7, plus 15 is 22, plus 20 is 42, plus 15 is 57, plus 6 is, plus 7 here again, that gets you 64. Woohoo! So 64 possible progeny, and at a ratio of 1/6, 6 5s, or 6 out of 64 chances to be 5 inches tall, and so on and so forth. Now keep in mind, this is the ratios that you get if you are heterozygous at all three. On the exam, or on any of your practice problems, and you should be quizzing yourselves, you might get heterozygous at 2 and homozygous 0 at the other, or homozygous 1 at the last. And you have to be able to produce for me the histogram that this makes. So hopefully this lecture gives you a bit of an appreciation of A, how beautiful genetics is, and B, the different modes of inheritance, from additive traits like height to control and color traits like our agouti baldness, to our simple mitochondrial inheritance and plastid inheritance, that's matrilineal, to our codominance and incomplete dominance mechanisms that are really subtle differences, but actually kind of the same thing when you look at them molecularly, to our X-linked inheritance patterns and our pedigrees. Genetics is quite beautiful. I happen to, study it for my PhD, so I have some biases here, and Punnett squares are kind of fun. Please try to give each other problems in order to practice your ability to solve different genetic questions. You will be asked to do so come time on the midterm. I will see you all next time for our discussion of linkage. Again, continuing on with Punnett squares, but introducing new variants to them, making it more and more an approximation of reality every time we go a little deeper.